As organisms age, one of the most critical threats to cellular health is the accumulation of DNA damage. This damage arises from internal stressors like reactive oxygen species, ROS, and external insults such as UV radiation or chemical exposure. If left unrepaired, it can lead to mutations, chromosomal instability, and ultimately, the transformation of normal cells into cancerous ones. Compounding the problem, aging itself weakens DNA repair mechanisms, allowing these errors to build up over time. Autophagy, though traditionally viewed as a mechanism for recycling cytoplasmic components, plays an increasingly recognized role in supporting the DNA damage response and maintaining genomic stability. At its core, autophagy acts as a quality control system, not just for proteins and organelles, but also for genomic integrity. When DNA is damaged, cells engage a coordinated network of sensors and repair enzymes to restore stability. However, when the damage is extensive or persistent and repair is not possible, autophagy can step in to remove severely compromised cells or components of the nucleus that are beyond salvage. For example, selective forms of autophagy, such as nucleophagy, can target fragments of the nucleus or chromatin for degradation, preventing the release of DNA into the cytosol, which would otherwise trigger inflammatory responses and promote cellular dysfunction. Furthermore, autophagy helps eliminate cells that have entered a senescent or premalignant state due to irreversible DNA damage. Senescent cells no longer divide but remain metabolically active, secreting inflammatory factors that disrupt tissue homeostasis, a phenomenon known as the senescence-associated secretory phenotype, SASP. By removing these damaged and dysfunctional cells, autophagy acts as a safeguard, reducing chronic inflammation and preserving tissue function. In this way, it not only prevents cancer by eliminating unstable clones before they can evolve into malignant cells, but also slows aging by reducing the toxic burden of senescent cells. However, the protective power of autophagy in the DNA damage response depends on its efficiency and regulation. With aging, autophagic capacity declines, which means that cells become less able to eliminate damaged DNA, faulty mitochondria, or senescent neighbors. This breakdown contributes to the accumulation of genomic instability and fuels both the degenerative aspects of aging and the pro-growth conditions favorable to cancer development. The result is a cellular environment increasingly marked by inflammation, reduced regenerative potential, and a rising risk of malignant transformation. Interventions that enhance autophagy, such as caloric restriction, fasting, and pharmacological agents like spermidine or rapamycin, have shown potential to mitigate these effects. By reactivating autophagic pathways, these strategies help rejuvenate the cell's ability to respond to DNA damage, clear senescent cells, and suppress early tumorigenic signals. Importantly, these effects are most pronounced when applied preventively or during the early stages of aging. In established cancer, enhancing autophagy must be approached with caution, as tumor cells may exploit these same mechanisms to survive DNA-damaging therapies. The link between autophagy, the DNA damage response, and aging highlights the body's reliance on internal maintenance systems to preserve long-term health. When autophagy is functioning well, it acts as an internal editor, constantly reviewing and correcting the genomic record, or deleting pages that are too corrupted to fix. When it falters, errors accumulate, errors that age the tissue, degrade function, and increase the chance of transformation into cancer. Supporting autophagy, therefore, isn't just about promoting cellular cleanliness, it's about protecting the very blueprint of life from the gradual erosion of time.